So this protein system that is composed of more than 20 plasma proteins, and most of these are lying as precursors of the enzyme. These proteins can be named as C1 to C9 and B and D. They are normally inactivated, but may be activated via specific pathways. We have seen that there are three mechanisms of their activation. One is antigen antibody complex, another is through the microorganism, and the B and D and propertin are associated with this one, and this is the alternate mechanism, and third one is the mannose binding lectin process. So whenever they are activated, their major function is to enhance the immune response and to eliminate the pathogen that is disturbing this pathway. So the classical uh, pathway is initiated by the antigen antibody complexes, and binding of antibody to antigen exposes a reactive site on the antibody's constant region. So this reactive site that is present on the antibody contact region interact with the C1 of the complement system and triggers the cascade of protein system and many of the enzymatic reaction. That this pathway then will result in production of many biological products that have got a specific tissue activity. So another mechanism, that is the alternate pathway or antibody independent, is directly activated by the bacterial polysaccharide or microbial surface. It bypasses the C1, and there is direct activation of C3, and C3 then is degraded into its constituents and leads to activation of the complement system and elimination of the pathway. Third is the process in which the mannose binding lectin MBL binds to microbial surface, and this binding of this microbial surface with the mannose will result binding of the microbial surface with the mannose binding lectin will result in the process similar to the classical pathway, but it does not require any antibody. So C1 will act on C4 and C2, that will degrade into C4-2B and C4A. C4-2B then this divides C3 into C3B and C3A. C3B then divides C5 into B and A component. Activated C5B then adds with C5, 6, 8, 9. So it makes a major complex C5B, 6, 7, 8, 9. And it is responsible for many of the action. So first is opsonization of the bacteria. The C3 binds to pathogen and mark them for phagocyte. That is the opsonization. Then they enhance immune cell recognition and clearance of the pathogen. So opsonization is by C3b, activation of the mast cells and basophils by C4-2b, C3a, C4a, C3a, and C5a. Chemotaxis is by C5a, and the formation of membrane attack complex is by this large molecule, that is C5b6789, that is responsible for lysis of the cell. So membrane attack complex creates pores in the bactericidal membrane that leads to osmotic lysis, and this is particularly effective in the bacteria that have got thin cell walls such as Neisseria. Agglutination is another biological activity by these complement system, and these protein alter the microbial surface so they are stick together, causing their easy clearance by the immune cell. These complement can also neutralize the virus by disrupting the structure of the virus, rendering them non-infectious. C5a, that is chemoattractant for leukocyte and another other immune cells at the site of the infection. C4a, 3a and 5a trigger the histamine release from the mast cells and basophils, and this then will affect the permeability of the vessel wall. There will be increased permeability and inflammation. So the inflammatory effects of this complement system will be increased blood flow and increased capillary permeability, and these promote protein leakage and coagulation in tissue to contain the infection. So any of the complement deficiency may predispose any individual to infections such as Neisseria, or if they are activated without any control, it may contribute to some diseases such as systemic lupus erythematosus. So if the particular complement is identified, the therapy may be targeted against that particular complement to treat the disease.